All right. Hi, everyone. Welcome and thank you so much for joining us at a virtual bite of science for Northern California. Also, thank you for joining me on a little bit of short notice here. We are, sorry, that's all messed up. So I am with the Center for Excellence in Education. We're a 501c3 education nonprofit founded in 1983 by Joanne DeGennaro and Admiral Rick Over, the father of the nuclear navy and civilian use of nuclear power. Our mission is to nurture high school and university scholars to careers of excellence and leadership in STEM. And we offer 3 cost-free programs to people in the United States. We have the Research Science Institute, the USA Biolympiad, and the Teacher Enrichment Program. First, we have the Research Science Institute, or RSI, which is a highly competitive 6-week summer science and engineering program open to rising high school seniors. It combines on-campus coursework and scientific theory with off-campus work in science and technology research. And this summer, like last summer, RSI will be hosted virtually. Our participants do really well at high school science competitions and often go on to advanced STEM degrees. Registration is currently closed. It opens up again next fall for rising high school seniors. Next up, we have the USABO, which is open to any high school students, and it's a series of examinations testing knowledge on biology concepts. The top 20 go on to attend national finals. I think that's actually wrong. And then the top 4, I'm sorry, this slide is actually quite wrong. The top 4 then go on to the International Biology Olympiad, which was supposed to be in Portugal, but will be virtual this year instead. There are many free student and teacher resources, which are a great prep for anyone just interested in learning about biology. And registration opens again next fall. And you don't actually have to worry about any of these dates because I will let you know and email you them. And lastly, we have the Teacher Enrichment Program. And we work to provide opportunities for middle and high school teachers to connect with leading experts from academia and industry, explore cutting edge research and development, and make some meaningful professional links with some benefits for you and your students. This year, we are running Virtual Bite of Science. We've reorganized our lab bench, and we have what we call public-private partnerships, but it's basically if you're interested in getting in touch with a researcher, a professor, someone from industry, you want to talk to your classroom, get in touch with me. I'm happy to help you. And I am actually very excited. So if you've been with us previously, you know that we have our lab bench website, and I'm actually very excited. We just launched our brand new website, and what I'm most excited about is Lab Bench is now on the TEP website. So as you can see here, they are divided by tab. We have life sciences, design and engineering, physical sciences, technology and mathematics, webinars and digital learning, and some additional resources. Under each tab are subjects within that. And so just to give you some help on finding exactly what you're looking for. And then today we actually have Michelle King, who's PhD, and she's actually the manager of the USABO here at CEE. And then after we'll hear from Kevin Stevenson, who's an associate engineer at Nordson Electronic Solutions. Any questions you have for presenters can be submitted to the chat box. And after our presenters are done, I'll hop back on and ask them some questions. All right, Michelle, you want to take it away?
Yes. Yeah, sounds great, Kim. I'll get this right eventually. Woo. Slides are good. Awesome. Thank you everyone for joining us at this TEP event. As Kim said, my name is Michelle King and I am the program manager for the USA Biolympiad. I will plug that one of the benefits of the program, in addition to studying, you do get to explore the breadth of the biological sciences, but it's also preparing these students for intensive college courses that they may not encounter a lot of the topics, or these courses are very large, and so they can't get the help that they need. So this is a really good prep for those students. So today I was invited to just give a little talk about my dissertation work, which was on calcium signaling network and Pseudomonas Aerogenosis. But I wanted to give you a little bit about my background and what actually got me interested I fell in love with research, or I knew the moment that I wanted to do research when my biology teacher, who had a physics degree, kind of got a little tired of my questions. He wasn't tired, he couldn't answer them because he didn't have the background. So he pulled out one of his textbooks and he let me read it. And then my question wasn't answered, and he was like, well, if it's not there, we just don't know. And I was like, what do you do when there's information you don't know? Do we just, live with these gaps in our knowledge? And he was like, no. There are people who research stuff and they are on the front edge of knowledge, and they help us learn, advance, and put the information that's in your textbook. And for some reason, I had never thought about that before. I always just assumed that knowledge was there, and I don't know how it's accumulated, but I was learning it. And as soon as he told me that, I was like, wow, I could be the first person to discover something. And that's one of the things I carried with me through my research is I love so much that I am now the first person in the world to know this very thing, which is very exciting to me. So that was one of the draws. And then as soon as I saw a bacteria being streaked on a plate, I was sold. Microbiology was my passion. I knew I wanted to study microorganisms, and I wanted to study like how they interacted with the environment. I was very interested in biofilms. So I was very lucky as an undergraduate student to begin research in a lab studying calcium signaling in Pseudomonas. And I continued my work in that lab through a PhD, because not only was my research fascinating, but I had found a mentor who inspired me, motivated me, and encouraged me to follow my passion. And that passion ultimately led to STEM education. So here I am, the manager at the USA Biolympiad, and I'm so thrilled. But I am excited to share with you my work. So let's get started. So just the picture of this plate, this is just Pseudomonas growing on an agar plate. So the green color is one of the things that it's most known for. It was actually diagnosed by the green color. So if you ever have an infection that's green, most likely Pseudomonas aeruginosa, and also that may not be a good thing. Because Pseudomonas aeruginosa is a gram-negative organism. So you'll see it on the image on the left. So that's an image I actually took using our microscopy facilities at OSU. So it's ubiquitous in nature. It's found pretty much everywhere. But what I think is the most interesting part of this organism is that it's opportunistic, meaning that you encounter it on a day-to-day basis, but you will not get a Pseudomonas infection unless you're sick or unless there's an open wound. So one question that I always ask myself is, how do bacteria know when you're sick and when you're not? I mean, it's not like they're at the doctor's, they don't hear your temperature. So that was one of the driving questions of our research, actually. And Pseudomonas also causes both acute and chronic infections. It is so diverse in the infections it causes. Usually microorganisms will infect certain parts of the body or they'll specialize, but Pseudomonas is just very talented and infecting everyone. And I'm apologizing because as a microbiologist, I love my organism, but I also hate my organism because unfortunately, it is a critical, in 2020, who characterized it as the second most critical pathogen to study because of its antibiotic resistance. And unfortunately, Pseudomonas is resistant to pretty much everything on the market. So they use combinatorial treatments. And unfortunately, we are getting into a phase where those will no longer work. So we need to find other therapeutics and think about what Pseudomonas uses to adapt to the body. And the more we learn about this organism, the more we can manipulate it and save these people. So I know that this slide is going to be a lot, but Pseudomonas has a number of virulence factors. And virulence factors are just these factors that help it cause a really nasty infection. So at the top, you can see kind of this transition between acute and chronic. And so an acute infection is seen as like a very aggressive, very rapid, you will see something, you will get very sick in a few days. And then a chronic infection is prolonged. So Pseudomonas, so in one instance, which we'll talk about in cystic fibrosis patients, Pseudomonas can infect them for pretty much their entire lives and be perfectly happy in this environment. So it's very important to study the switch that goes on. And if you look under the slide, you'll see a whole bunch of information. But what it's showing you is that these are most of the networks that are characterized now that have been shown to cause this switch. And at the very bottom, you'll see that's basically the result of the virulence factors. So we have the type 3 secretion system, which is attributed to an acute infection, and it secretes toxins. You have secretion systems that secrete proteases, so proteins that degrade other proteases, lipases that degrade lipids. And then, of course, flagella, which the bacteria use to move, is very important in acute infection because they got to get around versus a chronic infection. They're a little bit more stable and stationary. And of course, something that fascinates me the most is the green color of Pseudomonas. And that is due to two very specific virulence factors, which one is a siderophore, and siderophores actually steal iron from the environment to bring back to Pseudomonas. And iron is required for life, and it's at such low levels in your body that there's a continuous battle between the organisms infecting you and your body to keep the iron present. And so siderophores are one of the mechanisms that bacteria can use to basically remove the iron from your body. And then, of course, I would be remiss to speak about chronic and not talk about alginate, PEL, and PSL, and those are all polysaccharides, which help the bacteria get embedded into this like thick, sticky mucus. So it's very hard for antibiotics to penetrate. It helps keep nutrients in. It basically protects the bacteria until they're ready to break open and spread and then just keep growing. And Pseudomonas, as I said, is ubiquitous. And this isn't bad for us healthy people. Unfortunately, for those, it is found, it's found in water systems. So there's some work to determine the best way to line the water systems to prevent biofilms from growing. So water systems like hospitals or in cities where hotels store some water like on the roof, they've found biofilms there. The middle picture at the top is medical implants. So those are hip implants, which the biofilms will definitely reside. Hot tubs, one of the most known infections that it causes is hot tub rash. And that's a very, very mild and usually because of unclean hot tubs. And of course, the biggest thing is how it resides in hospitals. And so in the picture in the bottom left, every place there's a little indicator is a place that someone touches, and there's a potential to pick up Pseudomonas and spread it. And what's very concerning is that if you see in the middle picture, I have soap and I have ultrasound jelly because Pseudomonas has been isolated from soap dispensers in hospitals and other public places. And there were some instances where the actual, like the manufacturer of ultrasound jelly had some biofilms growing on one of their instruments. And so it actually contaminated a lot of this jelly. And so it wasn't as bad for like pregnant women, but for people where they use ultrasounds like for wounds and stuff, they were getting infected. So that's also a major problem. And then of course, when we talk about hospitals, we talk about how does it go from patient to patient? They're able to track them in wings, and they can track down to the nurse or the doctor who interacted with one patient or one reservoir, like the sink system shown in the bottom right, and then they go to connect with the patient, which of course, again, is very dangerous when we're in a hospital. So one of the primary environments that Pseudomonas is studied as a pathogen is within the lungs of the cystic fibrosis patient. And cystic fibrosis, for those of you who may not know, which a lot of people don't, unless you have somebody close to you who suffers from this genetic disorder. So it's a genetic disorder in one of the chloride pumps in your cells. And this disorder, it causes a number of different things: mislocalization, degradation. It depends on the mutation. There are several different mutations characterized with cystic fibrosis. But the end result is an imbalance of ions. And so you can see with the second cell there, so it's talking about acidification and depletion of water. So the mucus in their lungs that is used as like a barrier becomes really thick and really heavy, and like ions aren't able to like freely move and be in balance. And so they're all kind of congested in these areas where the thick mucus is. And plus cystic fibrosis individuals, and there's a myriad of problems that they have due to these few mutations, but their cilia doesn't work. And cilia and your lungs are extremely important to help clear out this bacteria. So you have a lot going against you. And what happens is that at a certain point in time in their lives, Pseudomonas is introduced to a cystic fibrosis patient, and then their morbidity and mortality significantly increases. So there's much more damage to their lungs. There are other bacterial fungal pathogens that do infect the lungs, but it is been correlated with Pseudomonas infection that causes the most damage and leads to the most sharp decline. So the goal of anyone who's a parent of someone with cystic fibrosis is basically to prevent them from getting this bacteria introduced to them, because once it's introduced, it does not go away. As I said, with Pseudomonas, it can live in the lungs of a patient for 60 years. And ultimately, it is actually the cause of their deaths because they slowly suffocate. And one of the ways that my lab comes in is, of course, we have this question is like, how does Pseudomonas get into the lungs of a cystic fibrosis patient and be like, I'm in the lungs of a sick person? How does it do that? It's not, of course, it's a bacteria. And so we actually think that it's this imbalance in ions that actually tells the bacteria, oh, there's an imbalance, something is wrong, it's a really good time to infect. And my PI, as with everything in science, did a mild mistake in her postdoc, and she added extra calcium to the media that she was growing Pseudomonas strain in. And she noticed that, or no, she forgot to add calcium. And so she noticed there was no green pigment. It didn't grow. It wasn't, wasn't as healthy. It didn't do what it expected to do. And then she remembered she didn't add calcium. But that kind of told her that there was something in a relationship between pigmentation, which again, that pigment is like really nasty to humans. So there's some calcium regulation going on. And so our lab looked at the impact of calcium on these virulence factors. So to look at the motility and flagella, we did what was called a swarming assay, where you just inoculate the center of the plate, and basically the condition where they spread further induces swarming. So you can hear that between 0 millimolar, which is no calcium, and 10 millimolar calcium, there's not only an increased diameter of the colony, but you see that green pigment. So there's definitely some calcium regulation. We looked at biofilm formation, and this basically the height quantifies how much biofilm there is. So there's more with 10 millimolar calcium. Those proteases I talked about at the beginning, more of them are made with extra calcium added. The pyoverdin, which is the siderophore I mentioned earlier, and another molecule, pyacyanine, which I studied, those were both induced by calcium. And I just wanted to touch base on pyacyanine because I worked on extracting it a lot as an undergraduate. And this molecule is redox active, which means it can enter a host cell and almost disrupt the electron transport chain. And it actually is very beneficial for the bacteria for a number of ways. So it's very important to look at what is regulating this as well. And then we performed virulence assays. And so there's a midrib, which is of a lettuce, which we injected. So PA01 is our wild type strain. So 0 versus 5, you can see there's an increase in disease zone. And then we also injected Galeria melanella waxworm larvae. And you can see that in the 10 millimolar condition, there was more death. So it does actually increase virulence overall, in addition to impacting these very specific factors. But why exactly is calcium so important? And I'm not sure like what your level of calcium signaling is, but until I entered this lab, I knew that calcium builds strong bones and then I needed to drink my milk. But I didn't actually know the role that calcium played in our cells. And this is a huge role. So our cells maintain a very low level of calcium. And then when it wants to do calcium signaling, it wants to signal your heart beating, it wants to signal apoptosis, it will increase the level of calcium and then decrease it. And that wave, that is a signal. And to our cells, they are so sensitive to the signal that the intensity, the amount of calcium that's in there, and how long that amount of calcium is in there will change how your cell responds, which is incredible. And our cells have a ton of different mechanisms to regulate this. So they have calcium channels, which allow calcium in. They have ATP-driven transporters, which will remove it from the cells. They have proteins that bind calcium and then either go perform a function or just bind calcium to remove it from the cytosol. And then of course, they also have calcium stores because you need some place to store all that calcium so you can increase it and decrease it rapidly. So that is like the ER. The mitochondria does have some calcium, but more so the ER has been shown to be the major calcium stores for cells. And in your heart, the circa, which is like a specialized ER, is also a storage for calcium. And that helps regulate your heartbeat. Calcium regulates almost everything. I encourage you to Google just videos where they visualize calcium signaling. I was watching one earlier around a wound, and you can see the calcium waves around the wound, which is signaling to the cells to start growing. When we are fertilized, when our egg is fertilized, it's actually a calcium wave around the embryo that starts life. So it's just, so calcium is incredibly important for us. But when my PI started her research, nobody had thought that bacteria could be capable of this type of regulation. Oh, sorry. Yes. So we have all this data, and we showed that calcium impacts all of these, external things, basically. And this cartoon kind of represents like what we knew. So we knew that bacteria actually have calcium transporters that are similar to the transporters in our cells. We know that bacteria have calcium binding proteins that are similar to the calcium binding proteins in our cell. And what's interesting is those calcium binding proteins are actually involved in signaling and not just removing the calcium. So there's got to be some kind of signaling component here in bacteria that we're not missing. And in an environment like cystic fibrosis, calcium is elevated. So this elevated calcium could be one of the major triggers of Pseudomonas that says that you are sick. And so we also know one very important thing, and this was shown in both E. coli and Pseudomonas. And if you look at the bottom where we have this little wave, so we measured intracellular calcium of Pseudomonas aeruginosa. And what we see is the point of the challenge is the bottom, and then there's a sharp increase, and then it goes back down to basal level. So bacteria also have this intracellular calcium regulation, potentially. Of course, we don't, and we never anticipate it to be as complex, but if you're living in environments where calcium is present and that's an important signal, it's important that we learn about it, especially for Pseudomonas. So our first goal was to look to see how Pseudomonas responds to calcium. So bacteria, to respond to extracellular signals, they have two component systems. The first component is a sensor that will bind to something, and the next component is a regulator, and that regulator goes and binds DNA. And so that's how they're able to regulate gene transcription. So our first question was, is there a two-component system that's going to be regulating this increase in these virulence factors in response to calcium? So in collaboration, we performed what's called a microarray analysis. It's where you can obtain all the mRNA in the cell and you wash it over this neat little chip, and you basically get intensities, and you can compare conditions. So we basically saw in the presence of calcium, Pseudomonas has over 62 component systems, and there was one that was upregulated in the presence of calcium. So we're like, this has got to be it, because two component systems, they're self-feeding, so the signal that activates them should be probably the signal they respond to. So we named it CAR-SR for calcium regulated sensor, calcium regulated regulator. And the next thing that our collaborator did was he made strains that did not have the regulator. So there's no way that the bacteria is able to respond to calcium. And there were two genes out of the entire genome of 5,000 plus genes, two genes were no longer regulated, and that is CAR-O and CAR-P. Yeah, so they were no longer regulated, meaning that these are the direct targets of this two-component system in the presence of calcium. And so my work studied CAR-P because we used a tool called iTASR to predict the three-dimensional structure. We actually predicted several calcium binding regions, which were very exciting to us because of course we want to look at calcium binding proteins when we're looking at the calcium signaling network. And it is predicted to form a 5-bladed beta propeller, and beta propellers have an incredible array of functions in bacteria and eukaryotes, which are very exciting. So we are very excited to follow this. And then one of the next things you do when you're trying to discover what a protein does is you look at its functional domains. And we can do this through comparing lots of sequences. So if one sequence is characterized to remove a phosphate group, if that sequence is found in 8 million proteins, then those 8 million proteins probably all do the same thing. Unfortunately for me, we didn't find anything that was actually helpful for the functional part. Just gave us some more questions. So it was exciting just to follow up on those. And in collaboration with one of the several undergraduate students that I worked with, we wanted to look at how conserved it was. Is it only found in Pseudomonas aeruginosa, which is like the main pathogenic strain of the Pseudomonads, or is it found in all Pseudomonads, including environmental strains that are non-pathogenic, because that can tell us like what role it's playing. So we looked at the currently available 2197 Pseudomonas aeruginosis, like all of the available Pseudomonads. We only found this gene in Pseudomonas aeruginosa. And there were actually 3 other species named, but we did an analysis to compare the genomes between Pseudomonas aeruginosa and these other, and they were misidentified as Protogens otitis and Mendocina. So those were all recharacterized as Pseudomonas aeruginosa. And the Pseudomonas SPs are strains that they haven't characterized down to a species level. And we actually, using CAR-P and this other analysis, we were able to verify that they are actually Pseudomonas aeruginosa. And this held true when we looked at all bacteria. There were a few samples that showed CAR-P was present and it was like a staph strain or something strange. But when we looked into the data, that specific sample was contaminated. So this is telling us something really exciting, that we have, it is not shared with eukaryotes, like some of our other proteins. It is not shared with other bacteria, like some of our other proteins. This is very specific to Pseudomonas aeruginosa. So the next part of his project and another project with another student that I worked with is we actually proposed the hypothesis that this can be used as a biomarker to identify Pseudomonas aeruginosa in clinical samples. Because right now, depending on your hospital, how much money they have, what techniques they use, most hospitals don't honestly identify infections, which as a microbiologist makes me very upset. But if a hospital can do a simple PCR and have a simple PCR machine, and this can be done by high school students, then you can actually test the isolates, and within 3 hours, 4 hours, if you got to count running a gel, you can actually tell if there's Pseudomonas present or not, where in the past it would take 2 to 3 days, or there would be very expensive techniques. And so this is an opportunity for like researchers and doctors to use CAR-P as a biomarker. But again, it tells us something extremely interesting, is that this protein is very specific for Pseudomonas, and it must be very important. So what we wanted to do, I'm gonna just admit I was working towards protein work. I wanted to crystallize it. As with all science, it did not go smoothly. So we kind of like, we went in a lot of different directions to just uncover as much as we could about this organism. And I'm sorry, I'm gonna like speed through this slide. But what we really also wanted to know is how specific is it to this calcium signaling network? We've actually seen that it plays a role in swarming motility. So when you have a mutant that doesn't have CAR-P, there's no induction of swarming, so there's no increased diameter, there's no pigment. So we were wondering if it plays a really important role, why would it just respond to one cue? And so we decided to perform a promoter activity assay, which is we utilize something that bacteria and organisms use, which is the Luxe Operon. And so the Luxe Operon produces light. If you're interested in that, I encourage you to read some about Vibrio fischeri and the relationship it has with jellyfish, because they use this light as like part of it's very interesting. So we use the Luxe Operon and we can put it in a plasmid. So we have a circular piece of DNA, and upstream of that Luxe Operon, we put the DNA region upstream of CAR-P. So that region upstream of the gene, as you guys all know, should be where everything is regulated. And so basically we can add a bunch of stuff and we will see if there's a reaction because there's more light, which is actually really cool. And I just kind of wanted to walk you through these techniques because I worked a lot with them over my past 3 years. So the most important thing for microbiology work going forward and doing any real experiment is normalization, because we have to kind of synchronize our cells. And that's actually really critical, because if it's done wrong, your entire experiment that you've spent 2 days preparing for is done. And so we normalize it. We give it fresh media with and without calcium, and then we put it in this really awesome instrument called a plate reader, or a Biotech is the actual brand that we use. And it's able to not only measure the light coming from the well, but it can also measure how much bacteria is present. So what we can do is we can normalize it, because you don't want when you look at the graph at the end, you don't want the increased luminescence just because of the increase in cells. You want to see that even with an increase in cells, you're still getting an increase in luminescence. So this graph just kind of shows you what the kind of data we'll be looking at. And so you can see at 6 hours, which is mid-log, there is a sharp increase in the light produced and then a decrease. And so that's a piece of data I will be talking about in just a second. So of course, our first question was like, does it respond to calcium? And if it responds to calcium, how much calcium? We know that a very high concentration of calcium induces CAR-P, but there's subtle gradients everywhere. And so we grew different promoter constructs and increasing levels of calcium. And as you can see by the graphs, when we add calcium, the tops of the peaks increase, and they do kind of shift too, which is really interesting. So that's saying that yes, calcium is increasing the production of CARP, but different concentrations of calcium may do it differently. So that tells us there may be multiple levels of regulation. And then we put it in a strain that doesn't have that transcriptional regulator, CAR-R, no calcium induction, which is what we anticipated. And we did what's called a calcium challenge, which was a very fun experiment. It's where I took it out at the middle of its growth or the very end of its growth, and I added calcium. And as you can see, the responses are significant and they're completely different. And that's important because at mid-log, it's in the middle of the logarithmic phase. That's where all the bacteria are growing and getting really fast and getting all the nutrients. And then stationary phase is where they're running out of those nutrients and they stop growing as fast and they kind of start to die off. So the fact that there are these differences is telling us again that there are other cues. And in terms of like so if you think back to acute and chronic, so when there's low amounts of bacteria, it's kind of acute. Maybe CARP is being regulated differently in these infections versus when Pseudomonas switches to a chronic infection and there's a lot of bacteria. So that was also very interesting. And this response isn't present without CAR-R. So that is the two-component system that is regulating this gene. We also looked at another important ion, which I told you was iron. So this is also imbalanced in the CF lung. And so we tested it originally as basically a way to prove reviewers wrong because they told us that calcium is not that important. There's like magnesium and iron and blah, blah, blah. We tested magnesium and zinc, no response, not special. Then we tested iron and we did get a response. And then our next question was, in these environments of the lungs, it's not just one ion or the other, it's both. You're going to be exposed to both elevated iron and elevated calcium. And our original thought was there would be no difference. It would just increase and that's it. But you can see from the green line, it increases and it keeps going and it goes until the end of the read. And we don't know when it stops, but this is just a huge activation that we did not anticipate in the combinations of these ions. So what we think is like, maybe these ions do positively regulate it, but there's other things about the environment that will kind of help temper this increase. Or CARP is just really important when there's elevated calcium and elevated iron. And then this response is not seen in the CAR-R mutant. So that two-component system is not just responding to calcium, it's also responding to iron, which is incredibly interesting. And next we wanted to look at, again, more of the role that it plays in the body. So we looked at the role of the response to the mutant in response to hydrogen peroxide. So our body uses hydrogen peroxide and other reactive oxygen species to kill off the bacteria. That's one of the major immune mechanisms. And you can see when we expose our cells with added calcium that the mutant that has no CARP, that's indicated with the little triangle, has less survival. So it's impaired somehow in this response. And again, this is all painting a picture that it is important for infections. And so we looked at its role in the Galeria melanella waxworm, which you can see in the top right-hand corner. And the elevated calcium is indicated by the black. So you can see that the mutant that has a little triangle in front of it is the percent survival is a lot lower actually than the wild type. And then when we put the gene back into the strain, it's restored. So this is telling us that there is some kind of balance. We know it plays a role in survival. It's extremely conserved, so it must be very important. But then there are these like subtle differences that don't really agree. And so there may be something larger here that we don't see because we cannot study in a human. That is illegal. And this is a little rough slide, but I wanted to throw this in because I also think it was really interesting. So we quantified the mRNA of CAR-P in our lab strain, and also 2 cystic fibrosis isolates. So that is the 31314A and the 22014B. And I'm going to just say that I would like to thank the Oklahoma Department of Health and the individuals who contributed these sputum samples, because it takes a lot to contribute sputum samples, and they do it for the betterment of our research, and we really appreciate it. And so you can see when looking at the transcription in these isolates, so yes, in CAR-P, it's elevated in response to calcium. Already know that. But in the isolates, what's interesting is that the first one has a significant response. So it's very significant compared to wild type. Second one has less of a response, which is, that speaks more to the heterogeneity within this environment and between strains within the cystic fibrosis patient. Because if you plate out Pseudomonas from a CF patient, you don't get one type of colony. You get multiple colonies. You get small colonies that will grow in 4 or 5 days because they're so slow growing. And then you get these colonies that grow immediately. And if you turn the plate upside down, the biofilm is so thick, it just oozes. So there's all this heterogeneity. And so we think that this is actually, we're going to follow this up and see what types of isolates these are to see if that could maybe explain the differences in response. Although all 3 strains showed an increase of CAR-P in response to calcium, which again follows our hypothesis. So just to sum it up, I unfortunately graduated before we could really determine the role of CAR-P in the cell. But I was fortunate to work with a number of other projects. So this image kind of shows you what we know about calcium signaling in Pseudomonas. So we know that CAR-SR responds to both calcium and iron. We don't know if it binds directly, but that's something a future study. And we know that this regulation regulates CAR-P. And then CAR-P is also regulated by oxidative stress and intracellular calcium, which we did not end up talking about because I helped characterize a putative calcium channel, which is actually responsible for generating that peak and bringing all the calcium in, so making the initial signal. So the CAR-P transcription was low in this mutant. So intracellular calcium is playing a role somehow. So we know that it's regulated by a lot of these factors that are very important for infections. It is found in every Pseudomonas aeruginosa that has ever been sequenced. So it's highly conserved. It's very novel just to this bacteria, but we don't know what it does. And fortunately, I have a whole team of people still working on it back in grad school, and we got to publish a few more papers. So all this work has been published in combination with undergraduate students that I worked with, and of course my wonderful PI and other collaborators. So hopefully one day we will know what CAR-P does, but I'm going to tell you when we find out, it's going to be really, really, really important. And so with that, as with any researcher, you would be remiss to thank the teams of people that helped you. I had an incredible set of collaborators, and my mentor to me is my female science icon. When I told her that I wanted to leave research because I thought there are bigger problems right now in STEM education, bigger than antibiotic resistance, bigger than these pathogens, bigger than the unknowns, is the lack of STEM education and the lack of appreciation for microbiology in high schools. And I'm like, I see this as the bigger problem that needs to be resolved, and I want to help. And so when I told her I wanted to leave the lab, I was very emotional, and she was like, you are passionate, you follow your dreams, and she supported me the entire way, and that's not normal. So I had the exception. And then, of course, I did have the privilege of mentoring a high school student. So you can see her accepting award in one of the bottom pictures. And then I can't underscore the value of having a good mentor and good lab team, and just you become a family, and I'll love all these people the rest of my life. And with that, I will take any questions.
All right. Well, I got to unmute myself, right?
Yeah.
Awesome. All right. So one question is, like, what do other propeller proteins do?
That's a really good question. So as I said, they're really diverse. Not only so they go from like 5-bladed beta propellers to 9-bladed beta propellers, and they can do everything like degrade sugars, degrade lipids. Sometimes they'll just bind to things and then perform a function, but a lot is in degradation. And then, of course, we looked at some beta propeller phytases, and that's actually where we're leaning towards. So phytases remove phosphate from phytic acid, which doesn't mean a whole lot unless you know what phytic acid is. And phytic acid is actually how, like, plants in our body store ions and inorganic phosphate, which is incredibly important. And so phytases actually remove the phosphates and remove the ions, and so that the bacteria can utilize them. So we're thinking that carpi has a role with phosphate and calcium. Yeah.
Awesome. And so you mentioned like that carpi is really conserved and also very unique to Pseudomonas, but that calcium signaling is not. So what are other sort of, I don't know, I guess conserved proteins maybe? I'm not sure what the like between prokaryotes and eukaryotes, like what are like some big ones?
Yeah. Okay, so I'm just, of course, talking calcium signaling because that's what I know. So there's one very specific protein domain. It's called an EF hand domain, and this hand coordinates calcium. And this domain is found in one of the most important signaling proteins in my mind in our cells, which is calmodulin. Calmodulin has 4 EF hand domains. And then calmodulin, once it binds calcium, it goes and binds other partners, and then that induces a major signaling event. So EF hands are found throughout eukaryotes and prokaryotes. Pretty much every branch of life has EF hand proteins. And so we identified one in Pseudomonas. The trick is to identify what it binds to, because we know that it's binding calcium, and that because it has this hand, that it's involved in some kind of signaling, but we don't know what the signaling or binding partner, and that's what was being investigated when I left. And then the calcium channel that I mentioned is actually very similar to a calcium channel that is in the ER that is regulated by pH, but interestingly enough, our protein doesn't have the residues that would make it respond to pH. So it's responding to something different, which is exciting.
Okay, so what do you think, what's your best theory for what carpi does?
Oh boy. So I mean, I've thrown out a lot of theories, like hypotheses for the function of carpi. Like I said, there is a lot of relationship between calcium and phosphate in bacteria. In E. coli, phosphate and some other molecules form channels that allow calcium in. In some other organisms, phosphate's used to store calcium. So bacteria don't really have a place to store except potentially the periplasm. So phosphate could be something. And then carpi could be degrading these to allow calcium in and regulating its function. It could also just buffer calcium, because our hypothesis is that while bacteria don't have organelles, the periplasm is actually how they store calcium for signaling events in the future. So carpi could be doing something related to that.
Oh, interesting. And then just as a, just so everyone knows, she's actually sitting across the hall from me right now. But I didn't know if you wanted to talk about anything with USABO for a couple of minutes, or share anything. It's exciting.
I definitely can. I'm incredibly excited. As I said, like, moving towards STEM outreach was my passion. I don't see enough kids interested in science because science is cool. I mean, it's fun. When I would work in Oklahoma with kids, they would have this very negative outlook on microbiology and they didn't want to study science because it wasn't for cool kids. And it just like really opened my eyes to like how we need to shift the communication in the topic. And so when I saw the USA Biolympiad, like, although it's a competition, and that's plain and simple, it is a competition. We want our team Biolympiad USA to go represent our country, you know, in the IBO and like win gold medals as with every other Olympiad. But what I love most about it is we provide students and teachers a way to start science clubs, start USABO clubs. We have resources that you can use to teach your students, but we also really want students to take the lead. And we have some clubs where you see students thrive. I have students this year that were emailing me and they had all of the information for their school and they're like, this is our proctor, but I'm in charge of the club, so I'm going to organize it. And I mean, it's just a joy working with these students who just, they feel so confident because they have these skills and they've been working and developing. And I also think with these clubs, it gives students the ability to teach other students. And the best way we learn is to teach others. So it's a really great experience. And it also brings together people who love science and just want to talk and learn and explore. There's still not enough microbiology in the Biolympiad for me. Actually, one of the topics for IBO is microbiology and biotechnology, so that's pretty exciting. But it's just so exciting to see how into it these kids get. I mean, if I had the opportunity to really, I mean, I came from a small school in Oklahoma. There are not many people, you know, primarily white. Just I had science teachers who were teaching out of field. You know, my classes were rather large because of budget cuts. We are like, Oklahoma's not known for state funding for science and education. And so just thinking about the impact that this would have made if I really had something to like give me the drive to study. And if I had been exposed to these topics a lot earlier, and of course it's all on me, I could have, you know, done Google searching, whatever. But if I had been able to like really explore and look at the material, like I've looked through the materials that we have in the USABO, and I like protein data bank, you can go and you can look at molecules of the month, and there's nothing more beautiful than a 3-dimensional protein structure to me. You know, there's all these cool resources that I think would have really helped me not only just like explore the passion and explore the field, but also like prepare me for college. Because when I entered into my intro to bio course and there was 350 people and my TAs were not bio TAs, they were TAs from other departments who just needed placement. And, you know, I was like, the topics were interesting, but it was hard for me to just follow and keep up at the fast pace. Because I loved my biology teacher, but he's a physics teacher. And, you know, he wasn't necessarily always on topic and, you know, X, Y, and Z. But it would have like knowing those concepts a lot earlier and at least hearing those words, I think would have like made me more confident in what I knew and like helped me absorb better. Because if you learn something at first when you enter university courses, you need to study 8 million times before you really absorb it. But if you hear it in high school and you're like already casually reading about it, and then you do some studying for this and you're in a club and you're like constantly talking, when you get to your intro to bio course and they're talking about population dynamics, you're like, oh yeah, I know exactly what happens to plants once volcanoes come through. I know exactly all, you know, plant anatomy. I got all this. It would make my freshman experience, which is jarring, going to college, first time you move out, first time you have complete freedom, it would have made it a bit easier. And so I really love the opportunity to give these students like, hey, study, have fun, take an exam, just one, you know, just see what it is. And then you really get to explore. And then our exams are written, they are not go to your textbook, read a definition, and verbatim repeat the definition. Our exams are designed to get students to think about how scientists see their data. So based on what you know, what do you think is going to happen? Or what technique do you think you should use? You know, so it's really trying to push kids into that mindset of like, you're doing research, you know, what would you see? What do you see from these results? Data interpretation. So that's something that I really loved coming from a high school of fill in the blank worksheets and just very, you know, matching. And that, I mean, that was all it. I'm not, you know, I think those are very valid teaching tools, but when you have teachers who teach out field and that's all they have to work on, and then most of your science teachers are coaches and history teachers are coaches, I kind of felt like, you know, I got left out of it. So I would have loved to have this opportunity, and I'm so excited to like be able to provide this opportunity for students.
Awesome.
And, yes.
All right, that's all we are to get to Kevin. Thank you so much, Michelle, for helping me out this week.
Yes, thank you.
And yeah, I mean, I'll see you around. All right, Kevin, if you are back.
I am.
Oh, awesome, welcome. I'm gonna mute myself, turn off my video, and take it away.
All right, well, let's see about sharing my screen here. All right, are you able to see my screen?
Not yet.
Ah, there we go.
Perfect.
We good now? Great. Well, I want to say thank you for the invitation to present. Again, my name is Kevin Stevenson, and I'm an associate engineer, mechanical in the key accounts group for Nordson Corporation, the Advanced Technology Solutions division. And up here on the front, you can kind of see, but I'll, let's see here. There we go. Little bits about Nordson Corporation. And for those of you who are maybe taking notes or something, I've purposely sort of listed bits and snippets and other things so that if you want to quickly jot something down or type something up, you can reference something quickly. But anyway, you can find out about Nordson by going to nordson.com. And I think the presentation will be available soon, and you can click on the link. But essentially, we engineer, manufacture, market differentiating products using dispensing adhesives, coatings, and sealants, and also biomaterials. Bit number 2 is therefore consumer non-durable, durable goods, and technology end markets. I've highlighted the areas in which my division is focused on. So dispensing adhesives, coatings, and sealants, along with the electronics. For the Asymtec products, surface treatment and using precisely controlled dispensing of the material, not only for location, but also volume. And here in Carlsbad, California, we design and manufacture both software, the machine, which is the electrical, mechanical, and the valves. So going back one slide, up here in the left-hand corner, that's a sample of our valve. Center photo, that would be one of our dispensing machines. So essentially, parts would convey in from one side, and the software would control both entrance of the parts. And you can see here in the middle, there is a valve, and we dispense fluids on mostly circuit boards and other things. So most of us have TVs and cell phones and computers. So the components and things that are attached that are protected in things like the voice modules, and cameras, things like that, chips on the board. The valves apply coatings or materials that will attach them to it. And essentially, bit number 5, convey it in, dispense it accurately, both placement, volume, and do it efficiently and quickly. So just some things about me and who I am. I I've been an associate engineer here at Simtech for the last 3.5 years. I've been employed here for 14.5 years, married 26 years. Today is actually my anniversary. I have a son and a daughter. My son is a junior in high school and my daughter is a freshman. Born, raised, and live in Southern California. Some highlights for Snippet 3, Eagle Scout. I have an AA in mathematics, a BS in biology, and an AS in engineering. I enjoy hiking and camping and most outdoor stuff. I'm also an avid CrossFitter, been an athlete for about 14 years and a coach for 6.5. Serving in the community, I coach sports for my kids in the neighborhood. I'm active in my church and I've done some outreach missions to Mexico, South Africa, and also Kenya. Experience, I've done construction, roofing, plumbing, framing, lots of other things. Teacher Credential Program, production chemist for working in a couple biotech companies. I've also done IT help desk and systems administration and as a mechanical technician as well. So what am I responsible for and what do I do at work? I work in the key accounts group. So our group specifically targets and focuses on our key customers and what they need. I monitor the machine configurations and their assembly exactly. I also test and validate for our customer specifications, the machines and all of the parts and assemblies that they want specifically on those machines in order to do what they want for their processes. So my key responsibilities for shipments are in the millions annually. The focus is to move from pilot, which is essentially existing or new products in development or new products that are being introduced, to actual production where other people follow directions and things to manufacture our equipment. And I've actually done that for both biotech and here in manufacturing. So what do I work with? I work with BOMs, which are bill of materials. I work with OMSs, which are operational material sheets. They describe how to assemble, set up, and test all of our assemblies and equipment. It covers both software, the mechanical aspects, electrical aspects, and our valves. Specifically, let's see here. This was kind of a big one, the work at number 4. So in order to do pilot to production, the interesting thing is that oftentimes with our key customers, we don't have anything that's actually released to production that's well formulated, well thought out, clear directions and things. So we actually need to manufacture and build in pilot and make sure that we can ship on time. But it's also creates a little bit of tension in being able to move things along to the production environment. So I kept asking myself, how can we make this easier on ourselves so that it's not always a lot of repeating last minute extra work and hard work for a few people just to make sure that we get things delivered, tested, and delivered on time. So honestly, using the scientific method, I asked myself, how can we improve? And through observation and some testing and other things like that, managed to be the architect to double production for shipments in 2019. I was a key contributor. We were still in pilot phase and was still creating the OMSs and doing training. And the result actually of doing that exercise was in the first 6 months of 2019, we shipped almost as many units as we did for all of 2018. So that was a really large accomplishment. And for that, my task at work is to do that for the new platform for one of our key customers, to move it from pilot to production, and also to integrate more, instead of separate little islands doing this, that, and the other thing, we're trying to integrate into one nice production integration. And again, responsible for the OMSs and training. STEM at work. I didn't know it, but yes, it is at work for me. So for instance, the bombs, the OMSs that I do at work, it really represents what I did in high school and in college in the laboratory classes. It's essentially lab experiments. And the key focus for me, both in biotech and here in manufacturing, is I basically need to write it and describe it so that someone else can actually repeat it and do what I did and get the same results. Like I said, lab experience, both biology, physics, chemistry, all of those played a role in helping me do what I do. For technology and engineering, those two items really help me to understand the big picture and the basics, and essentially to make connections in order to solve problems. And because there's usually, even though we try and do things reliably and repeatably, there are always items that come up that things don't always work the way they're supposed to. And so again, STEM number 3, again, the scientific method really helped my goal. And actually when management asked, you know, how do we increase production and how can we meet our customer demands? I'd already been thinking and kind of experimenting with it at work by observation, looking around to understand how, what kind of connections we have with people and the systems here at work, and really honestly asking the question over and over again, how can I? So again, I utilize pretty much all of my STEM training and basics, because when things do not go as planned, whether in pilot or actually production, or they don't go as well as expected, like I said, I'm constantly using the scientific method, and oftentimes it's interesting at different points. If you're keeping an eye out for it, you can make connections that you didn't know were there. One of the questions that was asked for being a presenter is what I do now, was that what I was aiming for in high school and college? And the answer is not exactly. So just a brief idea of how did I get here. I really didn't know what I wanted to do for my life when I was in high school. So I had questions and I didn't have a whole lot of money. No one in my family had actually finished college. So I didn't want to spend a lot of money to 4-year to try and figure it out because I didn't have it. So I went to community college and took classes and experimented, and decided that I like math, and I thought it would be cool to build bridges. So I had decided to become a civil engineer. In order to get to my 4-year, I worked construction and continued with some classes at community college. I eventually went to Cal Poly Pomona. Like I said, engineering was my goal, but some circumstances and things came up, and that didn't work out. Pursuing a math degree, that also had some challenges, and to be quite frank, focusing on work and earning the money to go to college, that actually hurt some of my my math skills. So I was looking at having to repeat some things. And so I decided with all of the math and science and things that I'd already had, a bachelor's in biology was the way I wanted to go. And thinking about it, you know, I really appreciated some teachers and some professors in college actually, who were kind of instrumental in encouraging me in things. So I thought about, well, I'll just return the favor and become a math and science teacher. So once graduated, sent out the resumes as most people do, got a lot of interviews, but no jobs. So wound up substitute teaching and starting my teaching credential program. And the teaching credential program kind of got interrupted because I was actually student teaching, or actually substitute teaching. And there were actually two teachers who had their PhDs. And I asked them why PhDs? Why did you leave biotech or research? And why are you teaching high school? And they said, you know, we like what we do and we wanted to enjoy it. So I thought that was really interesting. And so when I got a phone call out of the blue for a job at a biotech company, I said, you know, what do you think? And they said, you can always come back, and that kind of work experience is really valuable. And having observed them in their classrooms and things, I really thought and saw and observed that it seemed there was a higher level of challenge and learning going on in their classrooms. So I went for it. Worked in biotech, got laid off twice because with products that are in research, when you get bad news on third phase clinical trials, it's time to conserve cash, and so people get let go. So I went from one biotech company to the other. I had actually made a good impression with my boss's boss at my first job, and when he found out that layoffs were happening, he called me up and said, "Would you be interested in working over here?" So there I went. And then after my second layoff in biotech, went back to construction, picked back up the teaching credential, was actually working on my student teaching. And again, a little something happened. Actually, not a little something, but went back to construction. And so with all the things that I had been doing and had learned, I managed to solve an important problem for a friend of my wife's. And he thought that was pretty interesting. So he actually offered me a job in the IT. So I started out with the help desk and then worked my way up through the beginning stages of being a systems administrator. And then from there, I started out as a temp here at Asymtech as a mechanical technician. Wasn't really my training, but again, with construction and STEM classes and things, I picked up enough to where I could actually be competent in providing some assistance. So the temp position actually worked into a permanent position. And again, I started out here as a mechanical technician, and one of the opportunities here at work was with a lot of experience and some education, you can get a promotion. So that's what I did. I went back to school. I got my AS in engineering from Palomar Community College, and 3.5 years ago, I was promoted to associate engineer. So why STEM and why not? My answer is why not? Because it does matter. STEM really has given me a framework to live and work and play. It's allowed me to transition and achieve. It's really encouraged my observation skills, and it's really allowed me to kind of check and test what really is, whatever I'm doing. What math taught me, it taught me discipline, order my steps and show my work, especially in what I do right now. That's really important. You know, if I'm working on something that hasn't really been developed, that's, you know, the engineers that design it and think about it, how it's supposed to go together, and it's not working right, well, going back and reflecting on things that I've done in the past and how we can make it work. Math also taught me to look for efficient ways and connections of what I know, and because in math, you can work the hard way and the long way, or you can put some things together and work smarter, not harder. What science taught me is to wonder. It's a big world out there, and it really is amazing how it all works together. And there are also a lot of details too, and they matter as well. For IT and technology, how computers and information systems work was a real eye-opener, and it was always really interesting to see how people interacted and used that technology. And as far as engineering goes, I'm still learning. So STEM, it's challenged me to know the basics so I could make and extend connections to bigger things or smaller things in order to get things done, whether that's work or really in life. It makes things simpler and faster and better, in my opinion. So if I could have some encouragements and lessons learned, for junior high, I really think that affirming and encouraging wonder in science should be a primary focus. And also the fact that make an environment that is comfortable and students feel the freedom to take some risks, because in a lot of ways we're discovering, and discovery is learning, and there's oftentimes no really right or wrong answer. It's just kind of answering the question, good, better, best. How little makes up the big, and how big possibilities there are for their life and their future. I think it's really important, again, show your work and your steps to solve a problem. I'm a big fan of note cards. I still have some from college, and I've encouraged my kids to do the same. In high school, the encouragement is really practice the basics and master them, because that really allows you to make things permanent. And in practicing, it helps you do it and do it well, because in a lot of ways, I think most people are really looking to stand out, and that'll help you do that. The other thing in high school is it is work. I think we all would like to have it easy all the time, but science is work, and sometimes it takes extra work. But I think I know taking the time and the extra effort in the short term reaps mid and long-term payoffs. So, and again, encouraging them to look for the connections within their subject, but also within other subjects. And it looks like I repeated myself. Yeah, continue to show your work or your steps to solve some problems. Like I said, it's just invaluable to me, whether in biotech, because really in biotech, the idea was you hire degreed individuals in order to develop the pilot process, which is manufacturing drugs or whatever it is in really small quantities. But the opportunity is you want to manufacture that, and go live. And so we're talking from microgram, milligram quantities to kilogram quantities. So when you're doing that, you're really looking to scale things up, and having things written down, being able to reflect on them, how well did it go, is an important part. And if they start when things are simpler, when things do get more complex and harder, like they will, you have some good habits to support your success. And I find it really isn't that easy to remember stuff. So point number 9, be present. I find in a lot of ways in manufacturing, sometimes in biotech, and just working with students in different areas, don't be in such a rush to finish, because you actually shortchange yourself. And practice patience. Again, not always in a rush to finish. Last slide, I believe. Life seems to be an ongoing word problem for me. It's like math. You know, what have I got? What have I been given? What is it? If I don't know what it is, then find out what it is. Like I said before, I often ask myself, how can I? How can I do it faster? How can I do it better? Is there an easier way? Is there a way to help someone else out? Because asking those how can I questions, I think it's, again, sparks the wonder, but also it sparks growth and an appreciation for learning and actually to be a lifelong learner. The other thing is works for me, both in biotech and actually manufacturing, work seems to be sort of an unending series of lab experiments. You know, I'm always asked, can you do this? Can you do that? The customer is always asking, can your machine do this? Can your machine do that? So again, always asking the questions, how can I? Also asking the question, did I get the result that was intended? Well, yes. Was it good? Is it better? Is it best? Well, if it's no, what do I do? Do I do something different, or do I try something completely different? Well, if the answer is sort of, what kinds of things can I tweak or rework? What kinds of things can I change out or do a little different? Or did I make a mistake the first time and I just need to try it again? And again, it's what I do all the time in the pilot mode for both software, mechanical, and electrical. Point number 12, essentially what I do at work, I kind of live in an area, and it actually relates to point number 13. I call it big box or black box engineering. You know, those are the guys who are really doing the design work, who are getting the hard questions from customers. Can you do this? Can you do that? How can we do this? How can we do that? So they're designing things and creating things. And then we're asking individuals who follow directions and assemble things and put it together. So that's kind of the white sandbox. I call that production. What I really see right now as an opportunity in manufacturing industry is there really are very few people who what I call live or work in the gray, which is essentially bridging the gap between the black box and the white box. And I know enough to sort of live and play in the black sandbox, but I also I've worked my way up from just assembling things and installing things to where I am now. So I kind of have an appreciation or idea what goes on in the white sandbox. So like I said, gray is where I see the most potential for growth and for jobs for individuals who pursue STEM careers. And the other thing I've learned is you really won't know what connections you need until later on. So again, if you are patient and observant, then you never quite know how you will see things that come up and the connections they provide. And lastly, relationships matter, whether it's subject matter or people to people, people in your environment. And like I said, STEM really has given me a framework to do the job I do, to do it as well as I do. And that's pretty much it. Thank you for your time. You are more than welcome to contact me. I think Kim has my contact info. If you email me, just put in the letters TEP in the subject line. That way I know kind of who you are. My email flags people who contact me from external sources. So I can get to that. Some people ask, do you have tours? Well, we used to. Currently not right now, given the environment we're in. And as I was preparing for the presentation, Nordson has a nonprofit where we sponsor different organizations doing community service, education, and other things. And so I was thinking I'll ask our administrator for a list of the organizations, because I think there's could be some nonprofits that we are in contact with that may be available to make other connections to offer some assistance in what you do in the classroom. So thank you very much. And the questions.
Thank you so much. If we were in person, we'd all clap for you. So I also really want to thank you for presenting. It's actually very, very exciting to finally have a Nordson speaker at TEP. We've just never been able to make that happen, so I'm thrilled to have you. So I guess a couple of questions that came up. You've had a lot of different STEM jobs and in a lot of different fields and areas.
Yes.
What would you say are I know you kind of covered it, but what are your 3 core skills that allowed you to really transition through a variety of career fields?
To be really honest, I think it was the scientific method. I mean, really, all the class, I love learning. I like knowing how things work. And again, the scientific method really offered me an avenue to discover, but also to say, how do I do this? And again, it took a while, but learning the basics for math, learning the basics for biology, learning the basics for technology and things, because I chose to take risks. At first, I wasn't very confident, but I wanted to be someone in my family who finished college. And again, to be honest, I worked construction and I did really good work. I could have stuck with that. But I also understood that that takes a hard toll on your body, roofing, framing, all that kind of stuff. And I saw people who were kind of more ahead than I was. So I could have invested in a truck and tools and things like that and developed my own crews to do things. But I thought, I'd rather work smarter, not harder. And I think that was probably the other philosophy that just sort of came about. And I think, like I said, math lends itself to it. There's a lot out there in all the subjects. And the other thing is, I really didn't make some of the connections until I was put in an adverse situation where I had to kind of ask myself, how can I? I had to figure it out. So again, the big questions were, how will I handle adversity? And my observation is with students and even with people in manufacturing today, some people are afraid to risk, and a lot of times people are afraid to actually work or put in the extra effort. And so, so anyway, there really is no substitute for hard work.
I know it. I've been yelled at a job for taking initiative before, which is the worst, right?
Yeah. Well, I won't apologize if you don't like
I know.
it. I'm over it. Like I said, thinking about it, a butterfly, how do you get a butterfly from a caterpillar? Well, what does the caterpillar do? It goes into a cocoon, right? So the chrysalis, if you are watching it trying to struggle and get out, well, if you help it out, you're actually not doing it any favors. It only survives and becomes a butterfly and flies away because it struggles. So, and that was kind of the thing is, it took a while to adjust to taking risks and working hard.
Awesome. There's another question here. Let me find it. So you mentioned like you've done tours, but you know, obviously we're in the middle of pandemic, and I feel for you in California, over in DC, because it's been, I don't know, what are we on, like month 14, coming into month 15? I don't know, I lost track at some point.
Yeah, I'm not keeping track.
Would Nordson ever consider doing like a virtual tour?
Could be a possibility. I think there is some technology and stuff that would allow for that. And I can ask about that.
Awesome. Yeah, that'd be really interesting. And I want to ask, I know sometimes there's the processes that get like a little, sometimes it's trade secrets. So.
Well, no, not really trade secrets, at least in the areas that we could probably
that you would actually show us.
Walk through. I mean, we have a standard production line. There's just lots of machinery and parts and other things, people working. So, but it is interesting how things are laid out, things are processed, things like that.
No, my dad did dairy production work, and it was always fascinating to go into the plants. Even though I didn't know what things were doing, fascinating.
I think one of the fun things that I did, we visited a Jelly Belly factory up in Northern California, and that was fascinating. How hard can it be for a jelly bean? Well,
food production it's a lot of work.
Oh my gosh, you
got to clean everything all the time, because otherwise people get really sick, and then the company gets sued. It's a whole thing.
Well, it was just interesting how they actually formed the forms for making the jelly beans, how they actually mixed up the stuff and baked it, how they sorted it, how they mixed it, how they bagged it. I guess it's a jelly bean, but has some pretty interesting processes that go into that.
All right, awesome. We are actually out of time. If I have any more questions, I'll be in touch, but I also very much want to we'll be in touch about, if you have other resources. I know I've been in touch with Sarah, but and then we can certainly be in touch about a virtual tour maybe.
Sure.
But I really want to thank you for presenting. It's really always really, really interesting to hear people who have done a variety of things in STEM and have really, very clearly thought about those things. So thank you so much for sharing your sort of career journey.
Sure. Well, it's pretty non-standard, I think.
Wonderful. And then it's only because where is it? So I certainly want to thank you, Kevin, for presenting. If Michelle actually had to run, but thank you both so much for presenting. We have thank yous from teachers. I also need to thank our sponsors, which for this event are the Akamai Foundation, the Amgen Foundation. I'm on the wrong thing, so it would probably help out. Oh, yep. Boeing, Hologic, the KLA Corporation, Illumina Foundation, Jacobs Engineering, Infineon, Lockheed Martin, Nordson, longtime sponsor of TEP, the Novartis Institutes of Biomedical Research, TC Energy, Vulcan Materials, and WestRock. For teachers, I'm actually now just me and TEP again, but I have some help in the office, so I will be sending you guys speaker contact information, let you guys know when the recordings of these presentations are up, and get you guys some additional resources. So thank you all for joining me, and I hope you have a wonderful evening. And Kevin, sorry, happy anniversary.
Thanks.
I hope you have a wonderful evening.
I will.
Awesome. All right. Good night, everyone.
All right.